Hello everyone. In this lecture video, we're going to be talking about, in this case, the sensation of vision. Okay, the visual pathway in this case, right? So there's actually a lot of structures that actually help us see things. Okay, and the interaction and everything is about light rays in this case. Okay, so what happens is, so the way that you actually get to see objects is through in this case, an interaction between, in this case, your cornea. This is the cornea, the pupil, which is the opening, okay, which is also in this case a property of the iris. The iris is actually the cold portion of the eye, the lens, and the retina in this case. The retina is the innermost layer of your eye, and this is actually where the sensory, in this case, where the rods and cones, which are the sensory cells for vision, is actually located. So there's two ways that you see things. So you actually can see distant objects or close objects. So when you see distant objects, because the distant objects are almost parallel, right, in terms of light rays, in this case, because in terms of size, the lens does not need to be curved. There does not need to be a lot of refraction. So the lens is a little bit more flat. And the reason why the lens is flat is because the ciliary muscles, in this case, are relaxed. Relaxation, folks, is not in, it's actually going out. And because of the ciliary muscle relaxation, their suspensory ligaments actually pull and actually flatten up the crystalline lens. Okay, so that is actually how you see far vision. And actually, this is a very piss poor like image, because what you're trying to do is you're trying to get this area here into the retina in the back of your eye. FYI, this is the optic nerve, the optic disc, which is actually the entry point for the optic nerve into your retina. That's called the blind spot, and the reason why it's called the blind spot is because there are no rods and cones in that area. Okay, in here you actually have an area called the macula lutea, and in the middle of the macula lutea there's actually a region called the fovea centralis. That is actually the area of the sharpest vision here, because that is actually the area in which you actually have the most cones in this case. We're going to be talking about rods and cones soon. So distant objects, ciliary muscles relax, the lens are flattened because you don't need to refract a distant object. All right. When you are looking at a close object, what happens is, actually, again, this is a very piss poor image. What happens is you need to refract that image more, so the ciliary muscles contract. When they contract, the suspensory ligaments become a little bit more loose, and the lens become more rounded in this case. All right, and then obviously that image then gets reflected in this case into the back of your retina, so that your sensory receptors, your rods and cones, can actually detect them. FYI, if you have problems with the curvature of your cornea, you will have problems with vision. If you happen to have problems with your lens, you will have problems with your vision. If your iris, okay, has problems with your iris, you actually will have problems with your vision. So the way that LASIK surgery works, folks, is actually they actually reshape the surface, in this case, the curvature of the cornea. Okay, so that just remember that one. If you have, in this case, cataracts, what that means to say, folks, is this just gets cloudy. So then they actually do like just regular cleaning for that case. They actually just regular surgery in this case. If you actually have problems with your lens as you get older, you can actually replace the whole lens. Okay, so the lens can actually be removed, and actually a new lens can actually be dropped, and through the magic of medicine, actually the way your body works, the ciliary muscles in this case and the suspensory ligaments reattach to the actual lens. All right. Hyperopia is when you can see distant objects, but you cannot see something close. Myopia is, okay, when you can see something close, but you cannot see anything far. All right. So that's actually some terms that just as an FYI. All right. So how does in this case, the mechanism of vision actually happen? So in order for us to know this, we actually need to know the organization of the retina. Again, the retina, folks, is the innermost layer, okay, the innermost layer of in this case, of the eye. And it's primarily made up of multiple types of cells, okay? So the way this goes is, this is the most external here, and this is the deepest, okay, if you want to think about it this way. Light goes in, and what happens is light makes contact first with your two types of photoreceptor cells, or your visual cells, called rods and cones. Your rods and cones are also making contact with bipolar cells. Right, and your bipolar cells in this case are actually making contact with ganglion cells. Bipolar cell folds and ganglion cells are actually types of neurons. Okay, and rods and cones are also a different type of neuron, but they're receptors in this case, kind of like a gustatory cell or an olfactory cell. Right, this is the choroid coat. This is called, then the choroid coat is actually the middle layer, and then of course you have the, in this case, the outermost layer called the sclera. Okay. So again, this is the organization. So FYI, when you're alive, your actual retinal layer is actually colored. When someone's dead, there's no ATP production. This actual layer is actually, in this case, translucent. Okay, colored for living people and non-colored for dead people. Right. Two types of photoreceptors, rods and cones. What's the difference between rods and cones? Well, the reason why they're called rods and cones, folks, is if you look at this area in which it actually is embedded into the layer, into the epithelial layer, into the pigment layer, this is actually looks like a cone, and that actually looks like a rod. FYI, there are more rods than cones. Rods work, okay, normally in this case, if you want to talk about this, they actually respond mostly for like dim vision. Okay, so when you during nighttime, black and white, okay, conditions, the rods are actually much more active. Rods and cones are when you actually have bright light. Cones discriminate between colors, right? Rods discriminate between black and white images, grayscale, color scale. You have three types actually of cones. You actually have blue, green, and red cones, and actually that is the cause of color blindness, which is normally for men. If you are missing, or if you don't have an appreciable amount of the blue cones, you'll be blue colorblind, right? Green, if you're missing green colorblind, so on and so forth. All right, so that's actually where this comes from. All right, again, more numerous, black and white, dim light, less numerous, blue, green, and red, color discrimination. FYI, this is actually in high concentration in the fovea centralis, okay? Less on the periphery of the fovea centralis, right, which is again the area in the macula lutea. So how does the mechanism of vision happen? So this is a rod, and in the dark, it tells you here, what happens, folks, is there's actually channels, okay, kind of like just like facilitated diffusion. And what happens, and actually this is not facilitated diffusion, this is actually active transport. What happens, folks, is in this case ions enter, and what happens is an impulse travels through the, like in this case, the rod, and the rod then releases a neurotransmitter called glutamate. And in this case, the glutamate, which is the neurotransmitter, inhibits the bipolar cell. So the bipolar cell is on the off position when it's dark. All right, and when the bipolar cell is off, the ganglion cell is not stimulated, and no impulse is transmitted through your optic nerve, which is cranial nerve number 2. Right? When you have lots of light, okay, what happens, folks, is this channel's close, and because this channel's close, there is no there's no impulse that travels throughout the rod, and that means that there's no glutamate. And when there's no glutamate, the bipolar cell is no longer inhibited, which means to say that a neurotransmitter then stimulates, in this case, the ganglion cell, and then this nerve signal then eventually travels, and obviously this is gonna go to your optic nerve, which is cranial nerve number 2. So big difference, okay? Darkness, this is on, this is off, that is off. Light, this is off, this is on, that is on. Right? Big difference, glutamate. If you have glutamate, off, off. No glutamate, on, on. All right? So I hope that makes some sense, all right? And if it doesn't, please again, I have virtual office hours, and I am more than willing to talk to you personally.